Thank you so much for inviting me to this wonderful session. My name is Taichi Goto, a postdoctoral fellow at the University of Maryland School of Nursing, the United States. Today, I'm going to talk about our research findings so far to explore biomarkers associated with the trajectory of wound pain. There is no conflict of interest to be declared related to this presentation. Here is the agenda. When I worked as a clinical nurse, I sometimes cared for dementia patients suffering from wound pain, and so I had difficulty in assessing pain. So I was wondering how I could support these patients. This was the first motivation to start my research trajectory. Therefore, my final goal is to develop an objective wound pain assessment tool for such patients who have difficulty in expressing their pain. In Japan, the number of dementia is getting increased, meaning the number of dementia patients with wounds is also increasing. As mentioned before, clinical nurses sometimes have difficulty in precisely assessing wound pain. This is because there is a lack of pain assessment tools specifically designed for wound pain. This leads to inappropriate use of pain management methods. Therefore, I have been focusing on wound pain specific parameters to develop the objective assessment tool. So here, I'd like to introduce one of my research programs that employed a reverse translational approach, which means findings and questions from a clinical study were addressed with an animal study. At first, we attempted to verify if infrared thermography could be used for wound pain assessment. So we reported venous leg ulcers with high temperature surrounding tissue showed higher pain levels. However, this was not informative of the mechanism of pain, which is important information to decide a pain management method. So next, we focused on biochemical analysis of wound exudates and found a Navigr's factor, NGF, level in wound exudate in venous leg ulcer patients was moderately correlated with the pain level. NGF may reflect inflammatory and nociceptive reactions of the wounds, which means it could be a good candidate for wound pain generation biomarker. However, the correlation was still moderate. This may be because pain is a subjective experience and can be easily affected by many factors in addition to local wound parameters. So we thought wound pain perception biomarker is needed in addition to the pain generation biomarker. Therefore, we conducted an animal study that focused on the stress signals that can reflect pain perception. So we focused on the HPA axis, which secretes ACTH and synthesizes glucocorticoids at the adrenal gland. Previous studies show that such glucocorticoids exacerbated neuropathic pain and induced mechanical aldynia. Furthermore, other studies recently reported the expression of ACTH receptors on keratinocyte, and keratinocyte could activate glucocorticoid. Therefore, we were interested in whether ACTH could locally affect wound pain through glucocorticoid synthesis. We hypothesized locally injected ACTH, which mimicked stress response enhanced wound pain in the full-thickness cutaneous wound model. So we injected ACTH into a wound of the model and recorded the non-offensive behaviors and collected wound tissues for downstream analysis. Before addressing the main hypothesis, we validated the wound model for this study. The hypothesis here was locally injected ACTH-induced corticosterone synthesis at the wound area. We confirmed the expression of an ACTH receptor, MC2R, and a downstream enzyme, 11-vator HSD1 that was needed to make corticosterone. Also, we confirmed the corticosterone level was upregulated in the wound area of ACTH-injected rats compared with the control. Therefore, we concluded ACTH worked at the wound site. Thus, we moved forward to test the main hypothesis. Here, we compared the nose-offensive behaviors between the ACTH group and the control group. As you can see, the mechanical withdrawal threshold in the ACTH group was significantly lower than that in the control group, meaning a local injection of ACTH enhanced wound pain. The next question was what ion channel was corresponding to the ACTH-induced wound pain. So we tested for mechanosensitive ion channels, TRPV1, of which Dr. Julius was awarded the Nobel Prize for his findings this year, TRPA1, TRPV4, and ASIC3. We injected one of their antagonists in addition to ACTH and observed nose-offensive behaviors. As a result, TRPV4 antagonist most effectively canceled ACTH-induced wound pain. Next, we tried to clarify the mechanism of ACTH-induced wound pain through TRPV4. We found ACTH injection upregulated CYP2B12 expression that contributes to synthesis of TRPV4 agonist 5,6-EET. We also found 5,6-EET concentration in the wound exudate was correlated with the nose-offensive behaviors. Finally, we also confirmed 5,6-EET enhanced wound pain in this model. Taken together, we found a mechanism of stress or ACTH-induced local wound pain. Here we suggest 5,6-EET may be a good wound pain perception biomarker candidate. And this mechanism may be related to the chronification of wound pain, so the next question is how pain starts in wounds. So we next employed an omic approach to investigate the temporal changes of biological reactions in the wound pain model. Despite the importance to understand the temporal alterations of wound pain to determine appropriate intervention and prevent the transition of acute pain to chronic pain, there is a lack of study that focus on this topic. This study aims to analyze longitudinal gene regulatory programs in the hindport carrageenan inflammation model and hindport surgical incision model, and to compare the longitudinal gene regulatory programs in the two models to identify wound pain-specific gene expressions. The first model was a carrageenan-induced peripheral inflammation model, which is widely used in inflammation and pain studies. We injected 4% carrageenan into the left hindport of rats, and then nose-offensive behaviors were observed. Also, we harvested the hindport tissues and analyzed them using RNA sequencing and in situ hybridization. The carrageenan injection induced hindport edema, mechanical odynia, and thermal hyperalgesia. And infiltrated immune cells were observed in the hindport tissues after the injection, which means the inflammatory responses were induced by the carrageenan injection. This is the overview of the transcriptomic profile of the inflammation model. We detected and analyzed 2,920 differentially expressed genes through the observed time points. So the next topics are the transcriptomic profile of the surgical incision model and comparison of the longitudinal gene regulatory programs in the two models to identify specific gene expressions for each model. For the surgical incision model, we made a single 1 cm incision on the left hindport of rats. Nose-offensive behaviors were recorded and the hindport tissues were collected for further analysis. In like manner of the inflammation model, the surgical incision also induced hindport edema, thermal hyperalgesia, and mechanical odynia. The microscopic images show the infiltrated immune cells. This is the overview of the temporal changes in the transcriptomic profile of the incision model. We confirmed the largest number of differentially expressed genes was observed at 1 day after incising, when the most severe thermal hyperregia and mechanical aldynia were observed. So here we compared the two transcriptomic profiles. We found 253 information-specific, 7,647 incision-specific, and 2,667 common genes. Today, I will focus on the common genes because they may include important genes related to wound pain processing involving inflammation. By comparing the gene expression levels and gene expression ratio between two models, the expression level was much higher in the incision model than that in the inflammation model, while the expression ratio of each time point to the control was similar until 1 day after each intervention. This scatter plot shows the similarity of the gene expression patterns. This purple area indicates the incision specifically upregulated genes, which we called incision distinct genes at each time point. Among them, the most differentially upregulated gene was NPPB, which encodes B-type nitrate peptide BNP. This was highly upregulated at 1 day after incising, while the expression level in the inflammation model is lower. And in situ hybridization images show NPPB location in purple signals, specifically at the epidermal layer with one of the BNP receptors in the yellow signals. The roles of BNP in the skin tissues are still understudied, but it is reported that the peptides and its receptor were associated with recruiting T cells in the skin and with keratinocyte migration, and also associated with angiogenesis in vascular endothelial cells. So, these results suggest BNP may have positive effects on wound healing as well as wound pain, which is the next question to be studied. Future plans. We are going to unveil the detailed mechanism of ACTH-induced wound pain. Now, we hypothesize that ACTH-induced wound pain is mediated by a crosstalk between epidermal keratinocyte and sensory neurons. Also, the next step of the omics research is to investigate the functions of the incision distinct genes, including NPPB, and apply this approach not only to the incision model, but also to the full thickness cutaneous wound model and chronic wound models. This allows us to identify wound pain and wound healing biomarkers and therapeutic targets. This is the final slide. These studies were conducted at the University of Tokyo, Japan, and the National Institute of Nursing Research, National Institute of Health, the United States. So, I'd like to express my gratitude to Professor Sanada, Dr. Nakagami, and Dr. Minamatsu at the University of Tokyo, who were my great supervisors and mentors for my doctoral research. Also, Professor Iwata and Dr. Shinoda at Nihon University, who provided technical advice for the pain research using animal models. And here in the United States, Dr. Saligan and Dr. Gill were my former mentors at NINR. And Dr. Manes, Dr. Ayadorola, and Dr. Sapio were my collaborators for the transcriptome research at NIH. And Dr. Dorsey at University of Maryland. She is my current supervisor. Finally, I would also like to thank the Japan Society for the Promotion of Science, who provided fellowship grants for my doctoral research and research at NIH. Actually, I cannot conduct these studies without their continued support. That's it for my presentation today. Thank you so much for your kind listening.